My research interest is in the study of alcohol addiction, more particularly about the molecular and cellular changes that result from consumption of alcohol. Now, in the UK, like in many other countries, we consume a considerable amount of alcoholic drinks. These drinks can have an effect on a number of our organs, including the heart, the liver, and causes a number of cancers. This is a major cost to the NHS and represents a large number of admissions every year. One of the results of drinking alcohol can be that it can lead to addiction. Now, we don't understand why only some people that drink alcohol would incur into this phenomenon of addiction, but we do know that consumption of alcohol can cause changes in the brain, and we believe that these changes can lead to addiction. Now, in order to study alcohol addiction, we have chosen a model that has been developed by a number of labs, which is the Drosophila fly or the fruit fly. This model has a lot of advantages because it's quite easy to handle in the lab. It has several behaviors that are similar to human behavior in terms of alcohol consumption, and we share a lot of genes with the Drosophila, and therefore we can do a lot of genetic studies. Another great advantage of a Drosophila is it's part of a philosophy of replacing, reducing, and refining the use of animals in research. In order to tell you a bit more about what we've done, I will let my graduate students tell you what they've achieved in their studies.
As part of my PhD program, I helped Dr. Stefano Casalotti to set up the fruit fly lab at UAL. We had to establish suitable behavioral assays, and I was able to reproduce a tolerance assay, which had been described in other laboratories. Tolerance to alcohol occurs both in humans and flies, and can be described as a reduction in sensitivity to alcohol following repetitive alcohol consumption. We wanted to understand why this happens, and we focus on a particular family of proteins known as the G proteins. These proteins are associated with signal transduction in the brain. I was able to show that one particular G protein subunit, the G-alpha-Q subunit, is produced at lower levels following chronic consumption of alcohol. These may have had an overall impact on the alcohol-related behaviors. I was pleased that my work was continued by other PhD students and was published.
When I joined the lab to do my PhD, I was excited to continue a project that Tega had already started, for which she had done a good amount of preliminary work on. I was particularly intrigued by the observation of reduced G protein expression in response to ethanol consumption. Now, first off, I reconfirmed these findings and then went on to look at the project from a slightly different angle. Now, one of the great advantages of using Drosophila melanogaster, which is the fruit fly, is that we understand its genetics very well. Also, a large range of these mutant flies are readily available, and importantly, by breeding different flies with different modified genes, it is possible to develop a fly mutant that has the characteristics that you need. In my case, I was able to develop a fly mutant where we could artificially downregulate the expression of G protein, which is exactly what we observed in flies that were exposed to ethanol. And we then took these flies and measured their tolerance, and we did observe that they did not display characteristic behaviors. So therefore, as we have published, we have been able to demonstrate that G proteins, specifically the G-alpha-Q subunit, does play a role in the development of ethanol tolerance.
I joined the University of East London to do my MSc and continue to do Masters in Research under the guidance of Dr. Stefano. We all know alcohol addiction has a serious impact on human health. My project was to investigate the potential use of pharmaceuticals to overcome alcohol addiction. I chose to study a drug called Naltrexone. This is an opiate antagonist clinically used to treat alcohol addiction in human beings. I did a preference assay wherein fruit flies are initially exposed to high levels of alcohol in their food and then given a choice between food with and without alcohol. Flies that were pre-exposed preferred to consume alcohol-containing food, and normal flies consume food without alcohol. However, this behavior was reversed upon naltrexone treatment, and flies no longer showed preference to consume alcohol-containing food. Naltrexone also had an effect on protein kinase enzyme levels, which is implicated in alcohol addiction behaviors. I was able to publish this data as it demonstrated that naltrexone does have an effect on alcohol addiction behaviors, and that fruit flies are indeed a good model for pharmacological studies of behaviors which are also seen in human beings.
I was interested in exploring neurotransmitter receptors that could be targeted to treat alcohol use disorders. My main area of research was exploring the role of the GABA-b receptor and the importance of this receptor in modulating alcohol addiction-like behaviours. During my Master of Research degree, I was able to demonstrate that the GABA-b drugs could alter developed alcohol-induced behaviours. With these results, I was able to apply for and be awarded a PhD research grant from the Society for the Study of Addiction, which has allowed me to continue my research into my PhD studies. In addition to using the tolerance assay previously developed by my colleagues before me, I have developed a conditional preference assay whereby flies are trained to associate a neutral odour with alcohol vapour and a second neutral odour with water. After training, when the flies are given a free choice to move either in the direction of the odour associated with ethanol or the odour associated with water, the flies will move towards the alcohol associated odour but not receive the alcohol reward. Targeting of the GABA-b receptor with specific drugs has been shown to alter this behaviour. To confirm that this pharmacological intervention is capable of disrupting this addiction-like behaviour, through breeding, I've been able to develop flies whereby the expression of the GABA-b receptor is reduced. These flies behave differently when exposed to alcohol compared to the control flies, and are able to mimic the behaviour of flies that have had drugs administered to them, showing that the GABA-b receptor is of importance.
So if you want to learn more about what we do in the lab, please get in touch with us.